I'm Len Freedman. I'm Len Freedman, and I'm a co-author of a retracted paper. It's true. It's true. Which is funny now, but it wasn't funny at the time. And actually, what I'm going to talk about is actually a perfect segue to Pierre's introduction. I'm going to talk about a little bit, and very briefly in 7 slides, this issue of data reproducibility, or if the glass is half empty, data irreproducibility, and how standards in particular can provide a positive way forward. So if you could advance to the first slide, Houston, we do have a problem here. And these are just 3, obvious vignettes of how this data reproducibility issue can impact. You have gigabytes of data being sequenced. I was going to say some greater number, but I didn't want to embarrass myself at MIT in terms of the amount of bases that are being sequenced. But how do we know that the data that's being generated is uniformly being generated on sequencing machines, analyzed, shared, and integrated? We really don't. There is a genomic alliance that has started, and our friends at the Broad Institute are deeply involved in that, but it's very early days. There's also obviously issues around the use of antibodies, analytical antibodies, therapeutic antibodies, antibodies that we've all used, or I used to use, in the lab, from the Santa Cruz catalog. Hopefully nobody here is from Santa Cruz antibodies. But if the antibody didn't work, you just ordered another one until it did. But that, I think, as antibodies are being used more generally for diagnostics and therapeutics, it becomes a real issue in terms of the kinds of standards that don't exist in that area. And then if you even get into the clinical realm, we've heard stories of clinical trials for any of a number of cancer drugs that turned out to be invalid because the cell lines that were used early on in the discovery aspect of the work were contaminated or misidentified. HeLa being the most, I think, well-known example of that. So if you, next slide. So this is, again, reiterating what Pierre said in sort of a higher level view. What can happen without standards? Obviously, research breakthroughs are invalidated. Published papers, like one of mine, face retraction. Ongoing studies come to an abrupt end. Valuable time and resources are wasted. The public trust, I think, I'm not sure the public is very aware of this issue, but it's beginning to seep into the media. So I think the public trust is endangered. And obviously, the overall opportunity to enhance global health is delayed or completely lost. So I think we have an opportunity here that the quality and efficiency of life science R&D in terms of really placing important standards as the other industries have done. And I guess one of the questions I will probably get into as a panel is that why the life sciences have been so slow to incorporate standards is something that is really, I think, at this point, absolutely essential. Next slide. So Stacy asked me, because I was the first speaker in the first session, to just sort of level the playing field here, and I'm a little embarrassed to do this because I'm preaching to the converted here. Most of you, I think, that are here have self-selected yourselves because you're interested in standards, but I thought it would be important to define what standards are before we move too much further into the day. So standards are an agreed-upon way of doing something. They can be obviously quantitative or qualitative, and they're used, as Scott alluded to, and we're going to hear in the third session, they're used in all kinds of professions and fields, although not probably sufficiently enough in the life sciences. And there are 2 general kinds of standards. There are material standards, well-characterized, purified biological material. That includes microorganisms and cells, and even materials that are derived from nucleic acids and proteins including antibodies. And then there are written standards, and these are consensus-based documents. I think Mary Lou will probably be talking about this, on specifications, procedures, processes that are involved in biological materials or systems. Next slide, Pierre. So I want to tell you a little bit, and hopefully you'll be hearing more about this in the upcoming weeks, of a white paper that we commissioned, that GBesi commissioned, and it's entitled "The Case for Standards in Life Sciences, Seizing the Opportunities at a Time of Critical Need." We commissioned an independent research organization to initially interview about 50 stakeholders across the life science community in academia, government, industry, et cetera, to prepare a landscape white paper on this data reproducibility issue. So it was really assessing the quality of R&D methodologies, primarily in basic research, and really identifying the opportunities, in particular, in how standards can impact a meaningful change. The challenge, as we learned from the research, is that there are obviously multiple factors that contribute to life science research and the pressures that are being imposed on that. And that's depicted in this funnel slide here. So you can see that in the middle there's the traditional research quality checkpoints that those of us who have had research labs and have been supported by extramural grants have put a lot of faith in, and for better or for worse. There's the peer review grant review process, which has many of its own issues, and I'm certainly not going to go into that in the time that's allotted to me here. There's the intra-lab PI review. It's essentially the lab meetings that the PI will have with his or her group every week at lunch or at breakfast. There are the manuscript peer review that takes place when one submits a manuscript for publication. There is the colleague replication. That is, if your work's important enough that people are actually interested to reproduce your work because of their own area of research, it better work. If it doesn't, then there is a alleged self-correction component that occurs. But obviously, this is not a fail-safe sort of quality control checkpoint. And I just want to throw in here that there are all kinds of pressures that are being imposed on life science researchers. And again, this is not unique to the life sciences, but I think it's worth mentioning. And again, we can discuss this. There's tremendous competition. There are tremendous pressures for funding. There's obviously increasing complexity and collaboration around multi-centered grants, translational research, CTSAs, et cetera. And along those lines on the bottom there's more of a need to focus on translational research. That's what NIH wants to see, and that's what PIs are feeling compelled to do. So these components all came into play in this white paper. Next slide, Pierre. What we found, and this in a sense recapitulates the slide, is that there are at least 4 different kinds of stakeholders that are affected by the inability to reproduce research. There's obviously the labs themselves and their host institutions. There's industry and investors. If you're a venture capitalist investing in a little biotech company, and you put all your marbles in one basket, and that doesn't work because the work was flawed from the beginning, then obviously there's a failed investment there. I think that pharmaceutical companies who are certainly in this town, right around the block here, investing big in academia, want to be sure that the academic research that they've now sort of outsourced from their own discovery research is going to be reliable and reproducible. There are obviously the funders, and we can start with NIH and work from there. And I think that there is a danger of the public perceiving this as becoming an issue. And then finally, and I think an important role, and we'll hear from Veronique later on the role of journals and professional societies in terms of how irreproducible research can impact them as well. Next slide, Peter. And I'm finishing up here. So we're hoping that this problem that's been identified can be addressed in a very positive way from the increasing use and awareness of life science standards. There's no doubt from the white paper that these sort of pervasive systemic factors that I just mentioned within the life science community are placing increasing pressure on the research infrastructure. And I'm pressure for funding, pressure to publish, increasing scientific complexity. The solution, we believe, is really the development and adoption of standards. And that the success for that requires collaboration and a unifying process for all the stakeholders involved. And really this meeting is sort of the first step along that pathway. Last slide, I hope. So a little advertisement for GBSI. We're a new organization. We're providing a global independent forum to advance life science standards, really through targeted policy, thought leadership, and educational initiatives. We're advocating the increased availability of harmonized consensus-based standards. We're advancing credible, reproducible, translatable results in life science to enhance research productivity and facilitate future innovation. And I'm positive this is the last slide. I told you that the case for standards will be completed and published. It should be widely available in early December, and we're going to provide all of you with an electronic copy of the case, which we're really excited about. You can stop in and see us. We have an actual office in downtown DC on 19th Street between K and L. There's our email site, and we set up a live tweet. I don't tweet, but if you did, it's 2013 Standards Revolution. So thank you.
Thank you very much, Len. And we'll come back, I think, to what you said. But first, I think we should hear from our other panelists in various places. And though there would be benefits to adopt the standards, those benefits are delayed in time and quite diffused. Whereas the costs are pretty immediate, very tangible, and felt sort of and fall on a sort of more narrow sort of constituency. And so as a result, it's hard to get our act together. So that's one class of explanation. The second class of explanation is that it might be good for the system as a whole, but it turns out to be bad for one particular class of stakeholder, or for maybe many classes of stakeholder. The incentives are in some sense not aligned. And of course, it could be a little bit of both. Mm-hmm. And so you've all made sort of a very eloquent case for more standards in biotechnology, but as we're seeing sort of adoption being slow and development of those standards being slow, what do you think is the leading cause of explanation? Would you lean more towards sort of the collective action problems or more towards the sort of misaligned incentives kind of explanation? I'd be very interested to hear your thoughts about that.
Is that for us or for the audience? Or both?
For you first, Len, and then for the audience.
I mean, I've been thinking a lot about this question. I think you and I spoke on the phone, and I don't have the answer, but I wanted to expand a little bit and pose the question: why does this seem to be particularly unique to the life sciences, or to sort of basic research biologists in particular, as opposed to other industries, which I'm really looking forward to hearing about later. What is it? And it may be, a little bit of both of what you said, but I'm sure having been a bench scientist, the sort of misaligned incentives may be part of it. And I've talked to my friends and colleagues about GBSI and what we're doing, and a lot of the feedback is, but standards to me mean stifling my creativity. I don't want to be bothered with it. It's expensive. just leave me alone and let me, fool around in the lab, and if I'm lucky, I'll get a paper in Nature or Cell or Science. So, I'm just sort of expanding on your thoughts, but I'm also curious to hear from our panelists and from the audience as to why this has sort of evolved into an issue, and it's now a lot more acute than it used to be because of the stakes, because of everything that's been discussed, why it's so particularly true of this particular sector of science and research.